session and also of the workshop is Peter Park from Harvard Medical School, who's going to be talking about his work in ChIP-seq. So thank you, Peter.
Thank you, Chris. So I am standing between you and lunch, but Chris assured me that I have half an hour, so I hope you don't mind staying just a few minutes past 1 o'clock. All right, so I'd like to tell you some of the things that we've been thinking about in terms of ChIP-seq analysis. And knowing that a lot of people here are interested in whole genome sequencing data, I thought I would talk about a method that we've developed that's applicable both to ChIP-seq and whole genome sequencing data analysis. So Raphael did a great job of introducing basic issues in ChIP-seq analysis. I'm not going to actually talk about that. Instead, I'll talk more about sort of overview of what people are doing with ChIP-seq. And why it's so important to study chromatin and these histone modifications. So I'll describe that in some detail. I'll talk about integrative analysis multiple ChIP-seq profiles, which is what we have to deal with for a lot of our projects. Okay? So a lot of the methods that have been developed so far are to analyze a single ChIP-seq profile, but we now are interested in combining those and correlating that with other data types. And then I'll talk about a new method that we developed called BigSeq. And that's useful for calling broad regions in ChIP-seq data, as well as for finding copy number alterations. All right, so not much more to say here, other than that that from transcription factors, you get very sharp peaks, generally speaking. Okay, so those are fairly easy to call. And there are probably about 20 callers out there already. And I'm sure there will be more in the future. For histone modifications, sometimes you get fairly sharp peaks, but other times the enriched regions are a little larger. So those are a little more difficult to tell. You cannot rely on these positive, negative strand matches, for instance, to detect those. Okay? So that's something that we've been trying to address. And of course, nucleosomes are not quite ChIP-seq, but we do MNase treatment and then profile those. Now, why is it so important? So you heard a lot about transcription factors in the previous talk, but if you've studied transcription factors, you know that there are lots and lots of binding sites in the genome, but at a given tissue, a lot of those are not used. Okay? So why are some used and others not used? Some of it has to do with these nucleosomes. So if your transcription factor binding motif is in this linker region where it's open, a transcription factor can come in and bind. Whereas if it's wrapped around the nucleosome, then it's harder for the transcription factor to bind. Okay, so profiling these nucleosomes, and we can now do this with the base pair resolution in different cell types under different conditions is a very interesting field. We're also interested in studying profiling histone modifications. So these nucleosomes are made up of histone proteins that have these tails, and different places along the tail can be modified with different chemical modifications. Okay. So why is it so interesting? Well, there are lots of examples. One thing that we study is ES cell differentiation. Okay. So as the cell differentiates, the DNA sequence is exactly the same. Okay. But there are obviously room for other things like transcription factors that drive these development processes. But there are also a lot of epigenetic information that's encoded in the packaging of the DNA in these modifications. Okay? So as, for instance, as the cell differentiates, when it becomes erythrocyte, okay, there are steps along here encoded in the epigenetic marks that allow certain genes to be expressed at some later time point. So this is something that you cannot tell by profiling gene expression. You need to profile these modifications. Okay. So there are lots and lots of examples. This is a hot field at the moment. Lots of disease processes. Many genes containing mutations in cancer, for instance, they target histone modifications. There's a bunch of papers recently on EGH2, which is a catalytic subunit of something called PRC2, which lays down these repressive marks, okay, to suppress transcription. Okay? And if you get a mutation in this gene, which is a histone methyltransferase that lays these marks, then there are cancerous processes that get started. Okay? basic regulatory mechanisms. I mean, what these nucleosomes do, what these histone modifications do, they're involved intimately in a lot of the basic processes. So one thing that I've been working on is dosage compensation in Drosophila, where we're interested in studying how an entire X chromosome in Drosophila gets upregulated. Okay, so that's a very important process, and it's a very kind of a interesting process, right? How is it that an entire not just a single gene, but an entire chromosome gets upregulated, okay, by approximately twofold? And with ChIP-seq and all these profiling, we've made a tremendous progress in the past few years, and it's a really beautiful story that I like to tell when I'm in front of a genetics audience. Actually, on my last visit to England, I met a biochemist at Oxford named Rob Close, and then he had a very interesting question that he wanted to address, and after lots of emails and phone calls and some sequencing at Oxford and also in Cambridge, Massachusetts, we published this paper earlier this year, CP-challenge recruit a histone 3 lysine K36 demethylase. Okay. So what we show in that paper is that these CP-challenge, and surprisingly little is known about what CP-challenge do, actually recruit a protein called KDM2. Okay. That's really a histone demethylase. Okay. So they bind in vivo to unmethylated CP-challenge and then get rid of these methylation marks. And we think that that actually marks some regulatory regions in the genome, and that this is one of the functions, I think, of CP-challenge. So there are lots and lots of processes where we need to profile histone modifications. So just briefly, ChIP-seq pipeline looks like this. I'm not going to talk about this other than that there's a lot of effort still going on in peak calling, but not so much in understanding, say, whether you've sequenced enough. Okay? So this is something that we've addressed in our earlier paper, but basically the idea is, as you sequence, are you saturating the number of binding sites, okay? Whether that's a transcription factor or something else, okay? And the factor of in practice, how many reads do people get? So it varies from 3 to, nowadays 10, 15, and it varies not for scientific reasons, but because it's how much one lane of Illumina machine generates. So if you look at data sets from 5 years ago, probably, or 3 or 4 years ago, 3 lanes, or 3 million reads, and now you have more. Okay. So it's actually a difficult issue, and what we find is that if you're interested in finding these peaks, okay, under a statistical significance threshold, what you find is that as you sequence more and more, you get more and more binding sites. Okay? You can only talk about saturation if you have some threshold for enrichment ratio. Okay? So this is actually, I think, quite similar to people that do disease association studies. If you have more and more patients, you're going to find more and more genes that are correlated with that phenotype. Okay? Except that those genes that you find as later on are those with small effect sizes, right? Small odds ratios, and that's what we find with ChIP-seq data. So we have a model where if you specify, I want all the sites with, let's say, 5-fold enrichment in my ChIP profile compared to the input, and this is how many reads we have right now, then we can predict based on some modeling how many reads you might need. Now, so that's all I'm going to say about the actual method, other than the peak calling for the broad regions that I'm going to talk about in the second half. I should say that a lot of times we worry about these technical variation, but there's something more important that we've had to worry about for some of our projects. And there was a question earlier about the antibody quality. And in fact, the biggest problem that we had to worry about in a lot of these projects has been the quality of the antibodies. Okay? So as statisticians or as computational people, we don't kind of worry about this, but in fact, that this is a major problem that we all should be worrying about. And so we have a consortium project on characterizing chromatin-related factors for Drosophila. And as part of this project, we've done a lot of ChIP experiments, over 1,000, and we try to characterize a lot of these antibodies. And it turns out that we find what others have suspected for other organisms, which is that for histone modifications, when we go through a series of validation steps, something on the order of 60% work. And for chromosomal proteins, something like 70%. So a lot of these are commercially, probably the majority of these are commercially available antibodies, and some we got by writing to individual labs. But this means that, without worrying about GC content and all these other biases, you have to worry about 40% of your data if you are just getting data from, let's say, some projects where they have not validated these antibodies. Might just be wrong. They might be cross-reacting with other modifications and such. Okay? And one problem is that people don't really agree on how to validate these. So when we go through a bunch of steps, and some of these are very time-consuming, we can't quite, the fact that it would be validated in one or two of these does not necessarily mean that the ChIP-seq profile is correct. So this is a difficult issue for the field, and people have tendency to just publish whatever profile they get if there's something interesting without really going through all these steps. Now, I mentioned that one of the more interesting questions now that we are doing these ChIP-seq experiments in large scale, is to really look at patterns of histone modifications across different profiles. Okay? So, these are the 4 histones that make up the nucleosome, and they all have tails, and there are various sites along these tails that can carry these chemical modifications. And of course, one of the questions is, what does each mark mean? I mean, we know maybe a dozen pretty well. Maybe we have some guesses for another dozen. But we don't really understand what all these modifications do, or how do they work together. Okay? And so one thing that we tried to do is we profiled as many as we could based on the available antibodies, and asked the question, are there chromosomal regions with distinct patterns of modifications? Looks a bit busy, but let me just walk through this. So this is work done by Peter Kurchenko in my group, and the data have been generated by this consortium called Model Organism ENCODE. This is the fly genome. Okay? In different colors, sorry it's hard to see, but they correspond to the 9 what we call chromatin states that we defined. Okay? So we basically, so this is chromosome 2R, 2LX, and so on. We try to annotate the genome based on its chromatin states. And each chromatin state corresponds to some combination of different histone marks. Okay, so we are calling this sort of chromatin-based annotation of the genome. Okay? So there is some machine learning process algorithm that goes on in generating this. There will be a paper by another group coming out fairly soon on this topic. Based on HMM, but other clustering type algorithms could work here as well. And so what we do here is we try to annotate these genomes and look for interesting patterns. I mean, why are there certain regions that carry the same type? How are they distributed? How are they correlated? And so let me just give you some examples of what we see. So you don't actually need to know what these things are. I'm just trying to give you an idea of what we're trying to do here. Let's say, for instance, the X chromosome, in fact, on my screen has a slightly different color because it has a lot of H4 K16 acetylation mark. Okay? This particular region corresponds to, so these are polycomb related factors. There are really lots of binding of those factors on those regions. And so I can look through and see where those chromatin states are. There are certain regions, so these are 2 regions that are, again, it's hard to tell, but there's an RNA-seq profile that are both expressed. But in terms of their histone modification patterns, they're quite different. Okay? And so on. This is a heterochromatin mark. On Drosophila, the 4th chromosome is actually very interesting. It actually looks a bit like heterochromatin and euchromatin at the same time, depending on which features you look at. So by profiling with all these histone marks, we can learn more about exactly why it carries certain features. So I just zoomed into that upper left panel. Okay, so different chromatin states correspond to different enrichment of different factors. Okay. So obviously, trying to figure out how many states there are is it's like trying to figure out how many clusters there are in a dendrogram. And we try to correlate. In fact, we have a huge panel of how each of these features are related to other genomic features. All right. Before I talk about the algorithm that I mentioned earlier, let me just say a word about how we do a lot of our ChIP-seq work. So not for our consortium project, but for a lot of my collaborators, we actually do a lot of helical sequencing. So maybe some of you have heard of this. Most of you probably haven't done a lot of helical sequencing because there aren't that many machines available. Anyway, so the machine is about $1 million, but for us it was actually cheaper to do our ChIP-seq work on this platform than Illumina platform. It was probably by a factor of 2. So this is one reason, but there are lots of other good reasons for doing it on this platform. So this is a single molecule sequencing. There's no amplification. Okay? So that means that we don't have to worry about artifacts from amplification. So when we profile the same biological sample on Illumina and Helicos, we in fact see some differences, presumably due to amplification. Okay? Very easy sample prep. There's no library here to construct. The way the sequencing works is that you have these poly T things stuck on the flow cell, and you just put poly A on your samples, and that's it. So postdocs love this, right? And you can actually do ChIP sequencing with very small amount of material. Okay? Typical ChIP protocols will require something on the order of 10 million cells. If you're doing some ES work, some mouse work, you can barely get like 10,000 cells. Okay? So this is a huge issue. And so the fact that we can do this with a very small amount of DNA is important. Now, it's got lots of problems as well, and one of them being because there aren't too many machines available, it's unreliable. Sometimes it gets broken, and it's hard to get it fixed and such. But this has been great for our work. All right, so in the remaining half, I'd like to talk about the algorithm that I mentioned for calling these broad regions. Okay? And in fact, I'll talk mostly about our application of this to whole genome sequencing for detecting copy number variation. Okay? Again, the problem of finding enrichment in ChIP DNA versus input DNA is similar to finding amplified or deleted regions in tumor versus normal DNA. Okay? And this method that I'll mention is the read depth method. Okay? So we've heard quite a bit in the last few days about combining this kind of idea with split reads and make pair read information, so that certainly needs to be done. So I'm going to just talk about this one aspect of it. The data set I'll talk about is actually just single end. I believe what happened with this sample was, well, first of all, it was done probably a year ago, but I think that the quality of DNA was such that they could not construct these make pair libraries. So the sample that we analyzed is this glioblastoma sample, it's a brain tumor, from the Cancer Genome Atlas. And this was sequenced at Baylor on solid and at 30x coverage. Okay. And the good thing about this was we could, in fact, compare this to Agilent 244 and SNP AFI 6.0 in Illumina array because they had been done as part of the pilot TCJ project. Okay, so how does the algorithm work? Well, the statistics problem, I feel compelled to talk about the method in some detail here, since we're at the Mathematical Institute. So I have some formulas here. But the problem is quite simple, right? You want to just scale along the genome, and you like to identify regions where you see more reads from the tumor sample compared to reads from the normal sample. So how do we do this efficiently? So there are lots of good algorithms that have been published for array CGH. In fact, we have a website where people can submit their array CGH data, and we'll run 10 different algorithms and give them the output so that they can sort of see what the overlap is. Some of those are quite good, but the problem is that they're very slow. Okay? So we had to find something fast. Okay? This work is mostly done by a statistician in my group named Ruben, with some help in implementation from Joe and Tamin in my group. Okay, so what is the model? So given a short read R that maps the reference genome, we have two pieces of information. Okay? Position S and the read type, whether it's tumor or normal. Okay? And we have this joint distribution of Y and S, okay, that we're going to use to construct a likelihood. Okay? So we're going to denote Q of S to be the probability of a read at position S being a tumor read. Okay? So the notation is a little, it gets a little complicated, but the idea is very simple. This is probability of the read. Given some read at position S, what is the probability that it's a tumor read? And then we simply construct this likelihood. Okay? And so to actually have a model, we actually have to assume some breakpoints. Okay? So, and we're going to simplify or we're going to modify our likelihood a little bit. So we're going to assume that we have breakpoints, tau 0 to tau n+1 on some chromosome. And we assume that this Q of S, the probability that the read is from the tumor sample at that position, is constant between breakpoints. Okay? That's a reasonable assumption, right? And then we just modify the likelihood. Okay? And so one set of breakpoint corresponds to one model. Okay? And so it becomes a model selection problem. Okay? And we use BIC, Bayesian information criterion, to select the breakpoints. Okay, so we simply put the likelihood into this model and gives us some formula. Okay. The algorithm is quite simple. I'll describe it, the implementation of the next slide. But we do introduce one parameter. Okay, so one problem that we found with existing algorithms is that there are lots of parameters that we have to specify, like the window size, and most importantly, but other parameters as well. So we do have a parameter that controls the smoothness of the final model. Okay? So if we want something, aberrations that we're more certain about, we just change this lambda to that degree. Okay? For those of you who, with more mathematical statistics background, who still have a soft spot for proving theorems that are completely useless, we do have an asymptotic result here. So if you have some read at a reposition in some SAT, the breakpoint SAT that minimizes BIC is a consistent estimator of the true breakpoint SAT. That is, it will converge to the true SAT in probability as the number of observations goes to infinity. Okay? All right, so how does the algorithm work? Okay? Very simple, and that's why I personally like it. All right? We bin the data into small regions. Okay, so for our 30x covered genome, we take that to be about 10. So we could go smaller, but I think bin size of 10 base pair is pretty good. Okay. And so those are our taus in our model. Okay. And then we try every neighbor, two neighboring bins, and ask whether how the BIC changes when we merge those two bins. Okay, so we compare those two models. Okay, so we do this until BIC cannot be further reduced. Okay, so at every iteration, we're just examining all the pairs. But once you combine two neighboring reads, you just need to look at that neighborhood again, right? While keeping track of what's happening with other neighbors. All right, so we run this algorithm. So let's take a look at a very large alteration, 1.6 megabases. So this is our tumor reads. Along this chromosome 7, normal sample. So we have matched normal for every tumor sample, usually peripheral blood, because it's very hard to get the normal DNA for a brain. And we look at ratios. Okay? So it's pretty easy to detect here. If you look at Agilent array, this is a 2-channel array where you hybridize tumor versus reference, normal versus reference. This is SNP6, where you have higher density than Agilent, but generally the probes are shorter, and so they tend to be a little bit noisier. Okay? But it's pretty easy to detect in all platforms. How about something smaller? Okay? This is a 14 kb region. Again, tumor normal ratios. It's pretty easy to tell. In fact, we have a lot of data points. Each point here corresponds to 1 kb average, but we have lots of reads here. So this is pretty easy to tell. But on arrays, no surprise, Agilent just only has one probe here. You can't catch that. Affy has some more probes, but can't catch that. Okay, okay. Now let's look at some smaller ones. So in this example, this is 110 base pairs. Tumor reads, normal reads. We look at GC content and mappability just to make sure that there's nothing funny going on in the log to ratios. Pretty easy to tell. Okay. 20 base pairs. Okay. Pretty easy to tell. Okay. I mean, again, all this depends on your depth of sequencing. So if you had 3x coverage instead, you'd have to multiply this by by a factor of 10 to see the same level, same depth, same number of reads. We actually do see a fair number of sites where we have increased log ratios, but in fact the reads in the normal sample in that region is different from the background. Okay, so this might be related to some alignability issue, maybe more repetitive regions, but these are really uniquely mappable, so I'm not sure exactly what's going on here, but we do see a fair number of these. We tried to validate a lot of these. So this is the size of the estimated CNVs. And so we have sequencing and qPCR, and we do fairly well. They're fairly concordant. So we have one where we get a very different number, but we don't have an explanation. But qPCR doesn't always work. So I think 1 out of this many is pretty good. So when we look at qPCR validation, again, same data on a scatter plot, it looks like without that outlier, our correlation is pretty high. So compared to other methods like split read or mate pair driven algorithms, we're actually able to estimate the actual copy number fairly well with this depth of sequencing approach. We can also do calculations based on some simulated data as to what CNV size with different copies would be detectable using this algorithm. Obviously, if you have high coverage, then you're able to, sorry, this is all kind of the same color in this picture, but we have high power to detect these. And as you go down lower, it becomes a little bit harder to tell. Okay, so just to summarize this, it's fairly simple. And the fact that it's based on this BIC criterion makes it fairly attractive, I think, because we do have it's not so ad hoc. Okay. It's fast. Once we have the aligned reads, we can do 30x coverage in 1 or 2 hours. Most of that is actually ordering the reads, and we have this one parameter lambda that we use to control. We can extend this to many different cases. So when we look at, we're actually using this to segment, just as the chromatin-based state map that I showed, segment across multiple profiles. All we do is just add up the likelihood. Another advantage, I think, is to derive error estimates. So we can put confidence intervals on copy numbers and boundaries. So we have, it's actually much more difficult than I initially thought, but we've figured out one way to do it using GIP sampling. Okay? And again, we should be combining this with other types. So just to thank the people that did the work. Peter Kurchenko is our lead for the mod encode project with chromatin data, and Ruben has been doing the algorithm work. So basically everything I presented here is unpublished. We're hoping to submit them soon, but we certainly would be interested in trying this on some of your data sets if you're interested, and collaborating. So thank you so much.